Hello everybody. We were talking about the visual system and visual signal processing. So in last class I talked about the retina, which is the most important structure in your eye. The reason is the photoreceptors, rods and cones are located in it. As you know that retina is only located in the posterior part of the eyeball. It is the innermost layer of the wall of the eyeball and is located only in the back part of the eye. So what happens, if you see, this is the back part of your eye, and the retina has two layers, right? The outer layer is the pigmented layer, which absorbs the light, prevents the reflection, and also provides nutrition to the neural layer, inner layer. Okay, so this is the outer pigmented layer, and then you have the inner sensory layer or sensory neural layer. Okay, and this sensory neural layer has a number of layers and different types of cells in it, right? So sensory neural layer of the retina, and what happens, you already know that you have the photoreceptors, rods and cones, right, in this layer, and you also know that rods are more active in dim light or dark condition, and cones are more active in bright light condition, right? And another layer of cells are the bipolar cells. You must remember from last lecture. So these are the bipolar cells, and then the innermost layer is the ganglion cell layer, right? So these are the ganglion cells. And also you remember the horizontal and amacrine cells through inhibitory neurotransmitter GABA regulate or modulate the signal. So you have also horizontal cells, right, and the amacrine cells. So those are different types of cells. Now the axons of the ganglion cells. Now I am going to talk about the ganglion cells, because in the photoreceptors and bipolar cells, no action potential is generated, only graded potential. We talked about that. So slow depolarization that does not reach the threshold, or that does not trigger the action potential, right? However, the action potential is generated in the ganglion cells, and when the glutamate is released here, that excites the ganglion cells, and ganglion cells generate the action potential. They should generate the action potential because the axons of ganglion cells are very long, and they take the signal to the brain, right? So the signal must travel long way, and the bundle of these ganglion cell axons together form the optic nerve. Okay, so optic nerve. cranial nerve number 2. And here, is the place optic disc. And this is the location. If this is the optic disc here, the optic nerve gets out from the eyeball, right? Now one thing is interesting, that the axons are unmyelinated until they reach the optic disc. So in the wall of the eyeball, the axons of the ganglion cells are unmyelinated, which allows the light easily pass through it. You just see here, I explained before that this is the way the light travels, right? So light must pass through. These are the axons, unmyelinated axons, okay, the yellow, and they form the optic nerve here, right? So if these axons are myelinated, then the light won't be able to easily pass through this layer of axons. Make sense? So since they are unmyelinated, light can quickly and easily pass. And as you know, that light first pass through all the way through the retina and hits the photoreceptor cells, which are in the outer part of the retina, right? So then the signal comes this way. Bipolar cells, you see, these are the bipolar cells and these are the ganglion cells here, okay? So all the axons of these ganglion cells bundle to form the optic nerve that exists through the optic disc. That's why you see the photoreceptors are missing in this area where the optic disc is. Also, through the optic disc, the blood vessels pass. Okay, so what happens here will move towards the brain, right? So the optic nerves go all the way to the geniculate body, so like this. So these are optic nerves, optic chiasma or optic chiasm, optic tracts. So long, right, from here to the lateral geniculate nucleus of the thalamus. Okay, so this is the lateral geniculate nucleus. Okay, so these axons of the ganglion cells must be very long, okay, to take the signal all the way to the lateral geniculate nucleus. Okay, and then from the lateral geniculate the signal will be taken to the primary visual cortex in the occipital lobe. We'll talk about that, okay, later. So that's how the optic nerve is formed. Okay, now if you see the retina through the pupil, sometimes you go to your ophthalmologist or eye doctor, they will put a drop to dilate your pupil so they can easily look through it. The reason they dilate, that they want to see larger area of the retina. So this is how they see the retina. Okay, and this retina is a healthy retina, and this shiny area where light is reflected more than other parts of the retina. Why? Because the pigmented layer is also absent here. No photoreceptors, no pigmented cells. So the light reflects. That's why you see the reflected light in the optic disc. Make sense? That looks bright and shiny. Uh, However, in other areas you have the pigmented layer, right? So they quickly absorbs the light, the pigmented cells. That's why you don't see the other areas are that shiny. Instead, you have to put actually light through the pupil to see this area. When you look at the pupil, you know that pupil is very dark. I mentioned the reason why pupil is so dark, because the light that enters through the pupil is being quickly absorbed by the pigmented layer of the retina, so light does not reflect, does not come out. Uh, that's why you see dark. So it sucks the light or absorbs the light very fast, and that's why that pupil is always dark. That's why you need to put the light through the pupil to see the retina. Uh, Now, this is the area you see called the macula lutea. We talked about this, and the center of the macula lutea you have the fovea centralis, where you have the cones in highest density, right? So if you see the fovea centralis, this area looks more bright reddish color. The reason you already know. You must remember the retina is thin in fovea centralis, right, like this. So this is the retina and this is the fovea centralis, where the retina is thin because some layers are pushed to the side. Therefore, you can easily see the blood vessels. So let me draw how the eyeball gets the blood supply. So this is the eyeball, okay, and this is the optic disc, optic nerve, right? And what happens, a blood vessel artery that is called the internal carotid artery, I believe you have heard internal carotid artery, okay, which comes from the common carotid artery in your neck, and a branch of internal carotid artery is the ophthalmic artery. Okay, so this is the ophthalmic artery, okay, and from the ophthalmic artery a branch, an artery that pierces through the wall of the optic nerve or covering of the optic nerve. So this is the optic nerve, and you know optic nerve has the connective tissue covering, and this artery pierces the covering of the optic nerve and enters into the optic nerve, and then through the center of the optic nerve it enters through the optic disc into the eyeball, and this artery is called the central retinal artery. Central retinal artery. Makes sense? Because it is going to the retina. And then it divides into two branches, the superior and inferior branches. Okay. So here you can see the central artery enters and divides into two major branches or main branches. Okay. Now the occlusion or any blockage in the retinal artery, the retina won't get the blood supply. So the retina swelling of the retina, right? Since the tissue will start to die, and also the retina is become opaque. Okay, and that is a clinical condition, and if that happens, when you look through the pupil, you'll look the retina, it will look like opaque and a lot of swelling in it. The color will change to like whitish color. However, one thing you will see that the fovea centralis, this area is still red. Why is that? Because you know that in this fovea centralis area, the layer of photoreceptors and ganglion cells are very thin. So the choroid is just under the retina, right? Which is the middle layer, you already know, choroid. So this is the choroid, and you will easily see the choroid where the blood is still flowing, because choroid is still intact, right? Only the retinal artery that is going like this is damaged or blocked. Makes sense? But the choroid is still intact. So this area you will see opaque. However, since the retina is very thin here, you will be able to see the underlying choroid, blood in the choroid. And that's why this area, tiny area, fovea centralis, will still look pretty red, and that is clinically termed as cherry red spot. I don't know if you have heard cherry red spot. Okay, even the retina is damaged, but still you can see red in the fovea centralis. Okay, now we will see the development of the retina, which is very interesting. Retina is the only structure located in the peripheral sensory organs that is a part of your brain. Okay, so how retina is located inside the eye, inside the peripheral organ, but it is a part of the brain. You must remember when we talked about the development or formation of the brain in the very early stage, embryonic stage, three vesicles or pouches are formed, right? This is called the, anybody remember, prosencephalon, the top one, right? And this middle one is called the mesencephalon. Just repeating. And the lower one is the rhombencephalon. Okay, so different brain parts come from these three vesicles. Now what happens, the prosencephalon, this part forms two separate vesicles like this, divides into two. The upper one is called telencephalon, believe E, and the lower one is called the diencephalon. Make sense? And the diencephalon forms two small vesicles like this, and these two vesicles actually form the retina. Okay, so let's see this part only. So this is the diencephalon. Okay, and these two vesicles are formed, and then the vesicles form cup or funnel like structures at the end, free end. Okay, and this part forms the retina. Okay, so this part forms two layers of the retina. So in this part eventually two layers are formed, the outer pigmented layer and the inner cellular layer, okay, of the retina. So this is actually the retina, how the retina is formed. So that's why retina is considered as a part of the brain. Makes sense? Although it is located in the outer structure of the body. Here you can see the fovea centralis, where retina is thin, ganglion cell and inner cell layers are pushed to the side. This is the real picture of the human retina. So you can see here. Here you see different layers of the retina, cellular part of the retina. So you can see this is the outer. So this is the pigmented layer, right? And then the cellular layer has the photoreceptors. Then you have the bipolar cells, ganglion cells, and these are the axons of the ganglion cells from the optic nerve, right? And also you have the horizontal cells, and these are the horizontal cells and the amacrine cells. Okay. Now we will talk about different types of ganglion cells. So these are the ganglion cells, you know that, right? And ganglion cells could be different types. We classify the ganglion cells based on the size and connectivity. That is the anatomical classification by the structure, right? We divide into 5 different types of ganglion cells. 3 are the main types of ganglion cells. One type is called midgate cells. Okay, another type is called paracel ganglion cells. Okay, another type is called the bistriate. So these are three main types bistriate cells. Okay, let's first talk about these three types of ganglion cells. Midgate cells are also called P-type cells. Paracel cells are also called M-type cells. Okay, and bistriate cells are also called K-type cells. Why midgate cells are called P-type? Because the projection of midgate cells go to the lateral geniculate nucleus, and in lateral geniculate nucleus you have a number of layers. So the layers of lateral geniculate nucleus that receive the signal from the midgate cells of the retina, midgate type ganglion cells of retina, that those layers are called the parvocellular layers. Make sense? Parvocellular layers of the lateral geniculate nucleus. So these cells receive the signal, or these layers receive the signal from midgate. That's why these cells are also called P-type, parvo. Okay, and from paracel cells, the projection also goes to the lateral geniculate nucleus. But which layers of lateral geniculate nucleus? The magnocellular. Okay, there are two magnocellular layers. They receive the signal from paracel cells. Okay, and that's why these paracel cells are also called M-type cells. And bistriate cells project the signal to another type of another layers. Those are called kino cellular layers of the lateral geniculate nucleus. Make sense? So these are kinocellular layers, and that's why the bistriate cells of ganglion cells, type of ganglion cells, are also known as K-type cells. Okay, and midgate ganglion cells, if you see, the size is small, small cell body, and also if you see the dendritic arbor or branches, small. Okay, and these cells mainly process the color. They are excited by the change of color. So color sensitivity is present in these cells, midgate cells. Okay, and the midgate type ganglion cells receive signal from only few photoreceptors, rods and cones. Not many. Okay, so from few photoreceptors. Paracel cells are larger. The cell body of a paracel cell is bigger than midgate cell, and also the dendritic arbor or branches are much bigger, and they are mainly activated by the contrast. So small contrast can activate these cells. So they are very sensitive to the change of contrast. And paracel cells receive signal or input from many photoreceptors. So each paracel cell receives signal from number of rods and cones more than the midgate cells. Bistrate cells, these cells are, the term came from size of the dust, so very small, and these cells have been identified much later than the midgate and paracel cells, so relatively new and are recently identified cells, and these cells are sensitive to color too. So color change and contrast. Okay, so these are three main types of retinal ganglion cells and their projections and some properties. However, there are two other types of cells. Those are not responsible so far we know those are not responsible for the processing of visual signal, but they are responsible for the other things. One type of cells, the ganglion cells, they project the signal to the hypothalamus. So from the retina, these cells, retinal cells, there are few, send signal to the hypothalamus. Okay, and retinohypothalamic fibers take the signal, and the hypothalamus is related to the circadian rhythm, because hypothalamus controls the other glands, including the pineal gland that secretes melatonin, and melatonin is related to the sleep awakeness cycle, which is the circadian rhythm. Okay, so that is not related to visual or processing or sight, but related to the circadian rhythm. Okay, and another type of cells, those cells are excited and activates the pretectal nucleus and related to light reflex, right? So these are not related to your sight or vision, but they are also present in the retina. They belong to the ganglion cell layer of the retina. So those are 5 different types of cells in the retina. Now, if you see the physiological response of the ganglion cells, based on the response or firing of the ganglion cells, we commonly divide the ganglion cells into 2 types. Okay, one type is called on center of surrounding and another type is called on off center on surrounding. Okay, so those are two types of ganglion cell. On off, just opposite, right? Off center on surrounding. Okay, by the names you can just guess why they are called so. So let me explain. Okay, so first let me draw the photoreceptors. Let's see how they work through the rods. As you know, rods and cones, they are activated differently, but the signal transmission is same. So these are the rods. Okay, and you already know, we talked about it, the bipolar cells. So these are the bipolar cells. Makes sense? Let's say 5 rods. So these are the rods. Okay, and you also know that horizontal cells are here. So let's just say that this is one horizontal cell here, this is another horizontal cell here, and only the horizontal cells are connecting these, present in these synapses only, but not present in these 3. Okay, only present in these 2. So no. Okay, now this is the ganglion cell. Okay, so the receptive field of the ganglion cell, I am just drawing the receptive field of the ganglion cell here. So just think that this is a ganglion cell with its receptive field. So this is the axon, optic nerve, right? So this is the ganglion cell receiving input from all these 5 bipolar cells. Make sense? Okay, now if I present light on the retina, so let's just draw here, this is the source of light. So light is falling on these photoreceptors of the retina. Okay, light. Okay, good. So now when the light will excite these 3 rods, okay, that will activate all these bipolar cells and will release neurotransmitter glutamate here, and that will excite the ganglion cell. But which part of this ganglion cell? The middle part here. Okay, so this middle part will be excited. Is it clear? And since the horizontal cells are here in these two synapses, they will be inhibited. These bipolar cells will be inhibited. Make sense? So inhibition here, excitation, excitation, excitation. Okay, so this part will be excited, and this part will be inhibited, the surround part, because the synapse here inhibitory. Okay, so this ganglion cell is termed as on center of surround. Okay, so now if I put light only here, just think that only in this part, that means only these rods will be activated, no light here or here, so that will excite this ganglion cell. No inhibition, only on part, the center part will be excited, right? So if you record the activity of this ganglion cell by putting the light only in the center part, what will happen? You'll see this is the resting, and you'll see depolarization, action potential, right? You'll see this. So you'll see the burst of firing or action potential. Okay, in this condition. Now if I only put light not in the center but only in the surrounding part, so these two will be activated, not the center part. So that means this ganglion cell will be inhibited. Okay, because no excitation but only inhibition. In this condition, what I will see hyperpolarization. Okay, so no firing. The ganglion cell will be very quiet. But one thing can happen after that, hyperpolarization. When it will go back, the light will be withdrawn, it will go back. At that time it is called rebound phenomenon, that will go a little bit up, and that can cause some action potential. Okay, at the end of the after withdrawal of the light, okay, from the peripheral part. If you suppress something for a while and then when you suddenly withdraw the pressure, it may rebound, will go back and go further more. So this is hyperpolarization. If you stimulate the peripheral or surround part, hyper, and this is depolarization if you excite only the center part or on area. Okay, so this is on center of surrounding type retinal ganglion cell. Another type of ganglion cells or type of response of the ganglion cell is called off center on surrounding, just opposite, right? Instead of let me do this now. If I put the inhibitory horizontal cells here in the middle part, but no horizontal cell in the outer part, then what will happen when I will put the light? The center part will be off, right? And the outer part will be on, because no inhibitory horizontal cells are in the outer part. So just opposite. So how the connections are in the retina, how the ganglion cell is receiving the signal from the bipolar cells, and how the bipolar cells are connected to the photoreceptors that determine the response of the ganglion cell. Okay, Remember, each ganglion cell receives input from many photoreceptors and bipolar cells. Okay, so that's how we classify the ganglion cells. Let me quickly go over. Anatomically, by looking at the size of the cell and the dendrites, we divide into three types. Midgate. These cells have small cell bodies, small dendritic arbor, and they project to the parvocellular layers of the lateral geniculate nucleus of the thalamus. That's why these cells are also called P-type cells. They're sensitive to color. And another type are paracel cells. Okay, the paracel cells are larger than midgate cells. Cell bodies are larger, and also the dendritic arbors are more, and they project the signal to the magnocellular layer of the lateral geniculate nucleus. Okay, that's why they are also known as M-type cells. And the bistrate cells are very small cells. They are also sensitive to color, and they project to the kino cellular layers of the lateral geniculate nucleus. That's why the cells are also known as K-type. They are also sensitive to color. Okay, paracel cells are sensitive to contrast, difference between the light and dark. And other two types of cells, there are some cells, only few, they send signal when they are excited by the light. They send signal to the hypothalamus and activates the secretion of melatonin and regulate the circadian rhythm. You know that in daytime, in light condition, we don't sleep. In dark condition, we feel sleepy. You know that, right? So the pituitary, the pineal gland cells get excited, interestingly, when we are in dark environment. So when the pineal gland is excited, when we are in dark, that release melatonin and induces sleep. Okay, and in bright light or light condition, the pineal gland neurons are inhibited. Okay, and don't secrete melatonin, and we stay awake. So this is something interesting. Then another type of ganglion cells that are not for the processing of sight or image, but responsible for the pupillary reflex. We'll talk about the pupillary reflex in next lecture. So let's stop here. Thank you.